In this last part of the lecture, we're going to talk about some of the general features of each of the macromolecules. Let's start with carbohydrate. If you translate this word carbohydrate, it simply means carbon water. And when you look at carbohydrates in terms of their structural makeup, they really are just a combination of carbons and water, or hydrogen and oxygen. So 3 of the 4 essential elements are found in carbohydrate. Carbohydrate performs a lot of different functions inside of cells, but one of the ones that is most important is that it provides the short-term storage of energy for the cell. Now, we need to learn some of the terminology that we use when we discuss carbohydrates, and I have some of those terms here: monosaccharide, disaccharide, oligosaccharide, and polysaccharide. We'll talk about each one of those separately. If you translate the word monosaccharide, it's one sugar. So this word saccharide is simply sugar, and we often refer to carbohydrates as sugars. A monosaccharide is simply one unit of carbohydrate. It's the simplest form, in other words, that we find carbohydrates in. And again, it's what we often call simple sugar. Glucose is a great example of a monosaccharide. And glucose, remember, is a critically important monosaccharide in living cells because cells use glucose as fuel to build this molecule called ATP. You can also find monosaccharides as building blocks of other critical molecules. Take, for example, the nucleic acids, DNA and RNA. Inside of DNA, there's a monosaccharide called deoxyribose. This is a 5-carbon sugar. It's a simple sugar, and it makes up part of the DNA molecule. Ribose is a very similar monosaccharide. It happens to be found in the type of nucleic acid called RNA. Now compare monosaccharides to disaccharides. This term di just means 2. So a disaccharide is a combination of 2 monosaccharides that are bound together with a covalent bond. Even though there are 2 monosaccharides here, we still call disaccharides simple sugars. So, for example, table sugar or sucrose is a disaccharide. The sugar that you find in milk, called lactose, is a disaccharide. Now, you may have noticed that these words, glucose, sucrose, lactose, they have something in common. They all end in OSE. When you see the name of a molecule ending in OSE, you can assume that it's a carbohydrate. Oligosaccharides you may not have heard of before. This term oligo means few. So an oligosaccharide has few sugars in it. In other words, somewhere between 7 or 10 or 12 monosaccharides joined together with covalent bonds. The reason we care about oligosaccharides is because they are often present on the surface of cells. Oligosaccharides help mark cells as either being part of an organism or not being part of an organism. They help the immune system distinguish between cells that belong in the body and cells that don't belong in the body and need to be destroyed. Polysaccharide translates as many sugars. So polysaccharides have sometimes tens or even hundreds of individual monosaccharides within them. And each of those monosaccharides is bound to the other with covalent bonds. These are what people often refer to as complex carbohydrates. If you look inside animal cells, one of the most common polysaccharides is called glycogen. Now, glycogen is made up of many, many individual units of glucose, all bound together. So glycogen is a good way for a cell to store glucose. And you find glycogen, for example, in skeletal muscle cells and also in the liver, two places where glycogen is stored in animals. Starch is a common polysaccharide found in plants, and it's a lot like glycogen. It's made up of individual glucose units, many of them, all covalently bound together to produce this molecule called starch. And just like glycogen helps the animal cell store glucose, starch helps the plant cell store glucose. Proteins are macromolecules that are composed of subunits called amino acids. Proteins are made up of long, long chains of amino acids that are joined together, again, with covalent bonds. Now, we give the covalent bonds in protein a special name. We call them peptide bonds. There are 20 common amino acids found in cells. For human beings, 8 of those amino acids are called essential. And what that means is that those 8 amino acids can't be built by the cell. The cell has to get them from a food source. So we have to eat the essential amino acids, but we can build the rest of the amino acids inside of our cells. And all of those different amino acids, all 20 of those different amino acids, can combine in different order to create all of the different proteins that are found in our cells. Protein is coded for inside of our genes in our DNA. So each of the genes that you have in each of your cells, each of the genes holds a code to build a protein. Proteins are components of a tremendous number of different cellular structures, and they also perform a tremendous number of cellular functions. Just for a very short list, enzymes are made out of protein. And of course, enzymes are critical to the functioning of cells because enzymes catalyze chemical reactions. They allow for chemical reactions to occur in real time, very quickly, much more quickly than they would without an enzyme. And enzymes, again, are made out of protein. The receptors that are found on the surface of our cells are made out of proteins. The antibodies that circulate in our bodies and help keep us safe from pathogenic infections, those are made out of proteins. So I could make a much longer list than this, but those are 3 good examples of critical proteins in our bodies. One of the most interesting things about proteins is that each and every protein has its own unique shape. Because it turns out that that long chain of amino acids that makes up a protein doesn't stay as a chain. In fact, the cell spends a lot of time and energy carefully folding that chain of amino acids into a 3-dimensional structure. So there's some terminology that we need to know related to protein. The term peptide refers to a relatively short chain of amino acids. And by short, I mean somewhere between tens to hundreds of amino acids, all put together in a long chain in a particular order. Each of those 20 amino acids will be combined in a particular order to produce that long chain that is a peptide. Now, there are also polypeptides, many peptides, in other words. Polypeptides are simply long chains of amino acids, and by long I mean hundreds to thousands. Most of the proteins that you find in living cells are polypeptides first. They are made of hundreds and even thousands of amino acids. When you use the word protein, what you're talking about is a mature, folded polypeptide. Now, I'm guilty of using the word protein when I should say polypeptide. I do it all the time. But the important thing to remember is that polypeptides have to be folded up into proteins before they can function in a cell. Because the order or the sequence of the amino acids in every peptide and polypeptide is slightly different, that means that each protein is going to be slightly different. Every protein has its own unique three-dimensional folded up shape. And the reason that matters is because each of the unique proteins will have a unique job to do. It will have a unique function inside of the cell. Proteins are a great example of that concept of form following function. A protein is only able to do its job in a cell if it has the right shape, if it's been folded up into the right shape. Anything that would affect the shape of a protein will also affect its ability to do its job. Remember, we said when it comes to lipid, there are actually 3 different types. There are what we call phospholipids. That's the type of lipid that is abundant in cell membranes. There are fats, or what's also known as triglycerides. And there are sterols. Now, fats, or triglycerides, are the molecules inside cells that provide for long-term energy storage. Remember, we said carbohydrates actually provide short-term energy storage. Carbohydrates are a way to store the sugars that will be broken down by the cell in order to produce ATP for energy. Lipids are different. Fats and triglycerides set aside energy over the long term. This is the kind of molecule that the cell is not going to access frequently. It's setting aside enough energy for a rainy day. Another important function of fats and triglycerides is that they help to regulate temperature in human beings. In other words, the fat in the body is going to help to regulate body temperature through homeostatic mechanisms. Fat also just helps to cushion the body. All of the organs inside of the human body are cushioned by fat. And finally, we have the sterols. Sterols, like cholesterol, are a really important component of the cell membrane, and they're also a component in hormone. Last, we have the sterols. Sterols are an important component of cell membranes. Think about the molecule called cholesterol. Cholesterol is found in animal cell membranes. And even though cholesterol gets somewhat of a bad rap in that some of us eat too much cholesterol and it can cause problems medically, cholesterol is a required molecule in the body. Your cells could not function normally without cholesterol. Cholesterol is also critical to the building of certain types of hormones, sterile hormones inside the body, including the reproductive hormones. Cholesterol is the starting molecule for the synthesis of estrogen and progesterone and testosterone. And finally, we have the nucleic acids. Remember, there are two types of nucleic acid in cells. Those include DNA and RNA. DNA, or deoxyribonucleic acid, is the type of nucleic acid that contains genes, and it's the genes that code for the production of protein in our cells. DNA is used for a process called transcription, and the DNA will be transcribed to make a molecule called RNA, which is the second type of nucleic acid. RNA is produced in the nucleus of human cells. Another term you should know related to DNA is this term genome. When you hear about the human genome, what that word is referring to is simply the collection of DNA that we find in humans. When it comes to RNA, or ribonucleic acid, there are actually three different types inside of cells. Three primary types. These are called messenger, or mRNA, ribosomal, or rRNA, and transfer, or tRNA. RNA is involved in a process called translation. During translation, the RNA molecules are used to produce protein. The messenger RNA is the form of RNA that is actually translated, actually contains the code to build the protein. rRNA and tRNA are also involved in translation. rRNA is part of the structure of the ribosome, and it's the ribosome that is essentially the machine that's going to perform this process of translation. tRNA is the molecule that binds to and carries amino acids to the ribosome during translation in order to build protein according to the code in the mRNA. Now, mRNA, rRNA, and tRNA are the three primary forms of RNA that you'll find inside of cells. But there are some other forms of RNA inside living cells. We're learning all the time about new forms of RNA. And it's likely that RNA will play a role in medicine as we learn more about it. When we look at DNA or RNA, they're large, complex molecules. They are macromolecules. But like the other macromolecules of carbohydrate and protein, nucleic acids are made out of subunits. And we call those subunits, or building blocks, or monomers, we call them nucleotides. So many individual nucleotides are joined together by covalent bonding to create a strand of DNA or a strand of RNA. A nucleotide has three parts to it. It actually contains a piece of carbohydrate, and we mentioned this a minute ago. The carbohydrate is called deoxyribose in DNA and ribose in RNA. It contains a phosphate group, which is simply a combination of phosphorus and oxygen. And it contains what's called a nitrogenous base. Now, it's the bases in DNA and RNA that most people have heard about. These are molecules that contain a good amount of nitrogen. That's why they're called nitrogenous. And there are actually 5 different kinds of nitrogenous bases in nucleic acid, depending on whether you're talking about DNA or RNA. So there's adenine or A, thymine or T, uracil or U, guanine or G, and cytosine or C. In DNA, we'll find A, T, G, and C. In RNA, there is no T, so we will find A, U, G, and C inside RNA.